Fitness influencers promise to turn your body into a fat-burning furnace without explaining how mass actually leaves your body. Burning fat is not a localized melting process. It is a strictly aerobic biochemical reaction, meaning it requires a steady supply of oxygen to proceed. This reaction happens in exactly one place, deep inside the mitochondrial matrix of your skeletal muscle fibers. To understand fat loss, we have to look at the exact mechanics of how these mitochondria utilize oxygen to dismantle lipids and convert them into ATP. This newly created ATP, or adenosine triphosphate, is the primary cellular energy currency your body uses to power forceful muscle contractions. From there, we can map how this microscopic mitochondrial process scales up to permanently alter your whole body basal metabolic rate. Basal metabolic rate, or BMR, is the baseline number of calories your body requires to survive at complete rest. Zooming in on localized cellular energetics provides the rigorous scientific framework required to demystify macroscopic fat loss. It shows us exactly what happens at the cellular level when you train. Your muscle fibers do not pull from a single continuous fuel tank. The metabolic fuel they select at any given second is dictated by your immediate physical state. Take low-intensity movement, like a slow walk. The energetic demands on the muscle tissue are minimal and steady. In this state, your cardiovascular system easily supplies ample oxygen to the working tissue. This abundance of oxygen allows the muscle to prioritize lipid oxidation, breaking down fat for fuel. Sprint up a hill or lift a heavy weight, and the physiological environment shifts rapidly. A physical bottleneck occurs. The mechanical demand of the muscle suddenly outpaces the speed at which your heart and lungs can deliver oxygen to the cells. Without enough oxygen to process fat, the muscle is forced into anaerobic glycolysis. It shifts to breaking down glucose, a readily available sugar, because glucose can be metabolized without oxygen. This dual-axis graph maps substrate utilization against exercise intensity. As intensity climbs, carbohydrate utilization spikes, while the lipid oxidation line plummets toward zero. Because exercise intensity strictly dictates the fuel source, high-intensity movement actually shuts down fat oxidation during the workout. This establishes a physical paradox. How intense anaerobic work ultimately leads to long-term fat loss. To resolve that paradox, we have to track the fuel itself, starting with a single inert triglyceride droplet stored in your adipose tissue. The process begins with lipolysis. This is a chemical reaction that cleaves the triglyceride into smaller molecules called free fatty acids, allowing them to exit the fat cell. These free fatty acids are released into the bloodstream, where they travel through the vascular system toward the active skeletal muscle. Once they reach the muscle cell, they hit a cellular membrane bottleneck. Free fatty acids cannot passively enter the mitochondria. They have to be actively transported across the double membrane by a specialized carrier system called the carnitine shuttle. Inside the mitochondrial matrix, the fatty acid undergoes beta oxidation. The long carbon chain is sequentially sheared into smaller 2-carbon fragments, known as acetyl-CoA. These newly formed acetyl-CoA molecules are immediately fed into the Krebs cycle, a series of chemical reactions used to extract energy. The explicit output of the Krebs cycle isn't energy you can use directly. Instead, it produces electron carriers, specifically NADH and FADH2, which accumulate in the matrix. These are molecules designed to transport highly reactive electrons to the next stage of metabolism. The physical fat mass has now been successfully dismantled. Its potential energy is carried by electrons, setting the stage for the final conversion into usable ATP. This final process takes place strictly along the inner mitochondrial membrane, where those newly generated electron carriers arrive. This schematic shows the inner mitochondrial membrane, highlighting a specific assembly line of proteins known as the electron transport chain. As electrons are transferred from one protein complex to the next, their movement powers a series of pumps. Protons are pushed across the membrane, creating a dense electrochemical gradient. This entire chain requires oxygen to function. At the very end of the assembly line, oxygen acts as the final, irreplaceable electron acceptor, catching the electrons as they exit. When oxygen catches those electrons, it binds with hydrogen protons to form a harmless metabolic waste product, water. This clears the end of the line, allowing the chain to keep moving. The stored energy from the proton gradient rushes back through a turbine-like enzyme called ATP synthase, rapidly generating ATP. This demonstrates the ultimate dependency of the system. The entire process of lipid oxidation is strictly gated by the presence of oxygen and completely stalls out in its absence. We can now return to the macroscopic state immediately following a heavy, high-intensity lifting session. The intense mechanical demand has created a severe biological deficit. The local ATP pool in the muscle is depleted, and the tissue is left with a massive oxygen debt. This triggers a clinical state known as excess post-exercise oxygen consumption, or EPOC. EPOC refers to the measurably increased rate of oxygen intake following strenuous activity. While you rest on the couch, the body reverts to aerobic metabolism. It uses the newly available oxygen to clear lactic acid from the tissue and restore cellular homeostasis. This area graph tracks the EPOC curve. Notice the sharp spike during the workout, followed by a long, slowly decaying tail. The energetic cost of recovery persists for hours after you've stopped moving. Short anaerobic workouts force a massive compensatory mechanism. To pay back the oxygen debt, the body drives continuous lipid oxidation during recovery. Zooming out to the full human muscular system in a state of baseline rest, like deep sleep, reveals a highly active network. Skeletal muscle demands a continuous supply of ATP, even in the complete absence of voluntary movement. There are two primary baseline sinks for this ATP. The first is maintaining essential sodium-potassium gradients across cell membranes. The second is fueling continuous myofibrillar protein synthesis, the biological process of building new muscle proteins to repair tissue. Because you are at rest, oxygen is abundant. A larger total volume of muscle mass requires a higher continuous baseline supply of aerobically processed ATP, drawn straight from your lipid stores. Your muscles do not have tiny mouths eating body fat. They are governed by the thermodynamic reality of cellular respiration. Increased skeletal muscle mass serves as a permanent, high-volume sink for mitochondrial beta oxidation. By demanding more ATP at rest, that tissue elevates your basal metabolic rate 24 hours a day.
Thanks for watching Body Science School. Please like, share, and subscribe the channel for more.